Okay, so this lecture is going to focus only on the cell membrane. In the last chapter, we did an introduction to the cell. We learned all the different parts, what they did, and somewhat about how they work together. In this chapter, we're going to focus specifically on the membrane and the membrane's role in controlling what enters and leaves the cell and how cells communicate with other cells. So we're going to start with a little bit of history. Okay, so in the early 20th century, they knew that the cell membrane contained phospholipids, cholesterol, and proteins. However, they didn't really understand how it was arranged. So if you look at this electron micrograph, you'll see that this is actually two cells side by side, and you can see basically two phospholipid bilayers. They thought, because it looks uninterrupted, that the proteins must fit somewhere on the surfaces, not stuck in the membrane, because it appears to be sort of an uninterrupted bilayer. So as technology improved, they were able to do a technique called freeze fracture. And that's what this is showing here. So what they were able to do is cool the membrane down enough that they could then separate the bilayer. And when they did that, they were then able to see that the proteins actually were stuck in the membrane in different areas, and that's where they got the fluid mosaic model. And what that really refers to, fluid, comes from the fact that the phospholipids are actually liquid-like. They're not solid. And then a mosaic is sort of a picture made of tiles. I always give the example, if you go to Disney World and you walk through the center portion of the castle, on the wall there's a mosaic of the story of Cinderella. You can actually see little tiles making up all of the pictures. So that's what a mosaic is, and that's why it's called the fluid mosaic model. The fluid is referring to the liquidy part, which is our phospholipids, and then the proteins form a mosaic. They're kind of randomly placed tile-like structures in the membrane. Okay, so the fluid mosaic model, first of all, we talk about the phospholipid bilayer. The hydrophilic heads, remember hydrophilic, water-loving, they point outwards towards the outer part, the extracellular matrix, and the second layer then they point in towards the inside of the cell. The tails are hydrophobic, they are water-hating, so they face each other in the bilayer. Also, remember we said it's called the fluid mosaic model, and that's because the phospholipids are unsaturated. Remember, that means they have double bonds, and whenever there's double bonds, that causes these kinks in the membrane, and that's where the fluid part comes from. In fact, if the membrane gets too cool and it starts to solidify, it won't work properly. It won't be able to do the jobs that it normally does. So it's important that it stays fluid. The proteins, there's different types of proteins we're going to talk about in a minute, but some of the proteins are extrinsic, and that means they are actually on the surface, and then others are what are called intrinsic, meaning they're within, and a lot of them are transmembrane, meaning they go completely through the membrane. They stick out on the two sides. And then in animal cells only, we see cholesterol. Cholesterol actually adds to, at cool temperature specifically, it helps to keep the membrane fluid. Alright, and here's a diagram of a protein in the cell membrane, and hopefully you recognize the alpha helixes. Remember, this was from our biochem chapter where we talked about how proteins had secondary structures, and it turns out, if you notice, that the alpha helixes are sort of aligned with the lipid portion, and that would mean that the alpha helixes must be hydrophobic. So the reason they couldn't see the proteins in the original electron micrograph is because if electron micrograph was staining, let's just say the phosphate part, it was staining the hydrophilic portions, then it would also have stained the hydrophilic parts of the protein that were sticking out, and it wouldn't have stained this part. So you wouldn't have been able to see the protein. It would have been invisible. It would have blended. This picture here is showing just a phospholipid bilayer. Notice that the phospholipids, they show them all bent, and this would have to be from an animal cell because here's your cholesterol. Remember, it's a steroid, so you have 4 fused rings, and that would represent the cholesterol in the cell membrane. This is just showing a diagram, different representations of a phospholipid. Back when we were doing biochem, we would have talked about it on this level on the right, where we actually see the phosphate here, and then the 2 fatty acids. Today, and from here on out, we represented just the a phospholipid would just be the hydrophilic phosphates, just a circle, and then the two tails are two sticks that kind of have bends in them. Alright, so this is just another diagram showing the complexity of the cell membrane. And here, this is kind of showing what we mean about the unsaturated lipid tails. If you notice how they would prevent this from becoming a solid. This is what would happen if they were saturated. They would pack together and the membrane would solidify. And then here, this is showing cholesterol stuck in the membrane, which would also help. It says at higher temperatures it's not really helpful. It can actually reduce the fluidity of the membrane, but particularly at lower temperatures, this would be really, really important in animals as far as keeping the membrane fluid. Okay, so the lipids, remember, the phospholipid bilayer is a liquidy layer, and so the phospholipids are not stuck where they are. So for example, if this was a phospholipid that let's say I'm going to color it in, what can happen is this phospholipid can move, and maybe later I find it over here. What would not happen is this phospholipid would not go through to here and then end up on the other side. And the reason why makes sense, because this phosphate is hydrophilic and this whole section here is hydrophobic. So there's no way that this hydrophilic part would be able to pass through here to get to the other side. Alright, and the other thing that we're going to see is that the outside and the inside of the membrane are not going to look the same as each other because they have different jobs. For example, the outside of the membrane, one of the most important things that's going on there is you're going to have receptors to pick up signals from, say, hormones in the blood. You're going to have ID markers that are going to identify this cell as belonging so it doesn't get attacked by white blood cells. Obviously, white blood cells are not going to be inside the cell. They're going to be outside, so your receptors need to be outside, and your ID markers need to be outside. So that makes sense. This next slide that I have was just a little, I guess, self-check. If you had a fish that lived in an Arctic environment, how might its cell membrane differ? And we talked about this in class. For example, we know that the more unsaturated fatty acids, the more likely a membrane would stay fluid at lower temperatures. So maybe it would have a higher concentration of fatty acids in the membrane. Another thing is, we said animal cells have cholesterol. And if it's an Arctic fish and it's living where it's very cold, it would be very likely that it would have a higher cholesterol content in its cell membrane than other fish as well. And that might change it as far as eating the fish as well. That, you know, when you're talking about certain things having much higher cholesterol concentrations than other things, this could be one of the reasons why, because just simply their cell membranes are adapted to where they live. Okay, now the most important thing in the cell membrane, honestly, is the proteins, because all of the jobs that the cell does, it's going to rely on what can get in and out of the cell. And for the most part, most things can't pass through the cell membrane, not through the phospholipid part. That's what makes it selectively permeable. Really, what's getting selected are things that can pass through, for the most part, pass through the proteins. So the proteins are going to determine what the job of that particular cell is. They are the most important parts. They fold up so that the hydrophobic and hydrophilic portions align with the membrane. And here we are, our first one is transport. So transport is what basically takes molecules in and out. Most molecules cannot go through the phospholipid bilayer, particularly ions, particularly covalent things that are large, or polar covalent things that are large. So, proteins, sugars, and then all of your ions, and those are things that a cell needs. So obviously the proteins are going to be the key. A good example of this is in cystic fibrosis. We've used this as an example before. Remember, it goes back to the DNA having an error, which then causes a protein to be made wrong. It then folds up incorrectly, and in this case, this is a protein that controls the passage of chlorine or chloride ions. And so if we look here, we see this is the normal channel protein folded up the proper way. Chloride ions can pass through. Here's the protein from a person with cystic fibrosis, and this particular protein is folded incorrectly because of a DNA mistake that then causes the wrong amino acid. So you get the wrong secondary and tertiary structures. And then when that happens, that person's not going to be able to transport the chloride ions, and because they can't transport the chloride ions, then you end up with this mucus buildup. So that's a really good example of the importance of protein membrane, or membrane proteins that keep the cell alive. A second function of proteins in the membrane is that they can behave as enzymes. We're actually going to do a whole chapter where we talk about enzymes. Enzymes speed up reactions, and so membrane proteins could function as enzymes that are speeding up reactions that are happening at the membrane. The third one is support fibers. Your book actually breaks this into two separate functions because, for one thing, you've got the fibers that are holding membrane shape, anchoring it to the cytoskeleton, remember microtubules and all of that. But you also have, in addition to that kind of support fiber, you've got the cell junction proteins as well. Remember desmosomes and tight junctions and gap junctions and all of that. That goes along also. Like I said, they separate it, but it's pretty much, to me, it's kind of the same function. It has to do with supporting and connecting. The next one is receptors. This is really important because these kind of proteins pick up messages from the outside and then cause a reaction to happen inside. Most hormones, unless they're steroids, because steroids can pass through the lipid bilayer, protein hormones can't. So what they're going to do is they're going to attach to a protein, sort of like somebody knocking on the door, and that stimulates you to get up and go answer the door, or to get up and do something. A fire alarm goes off and you then respond in some way. So that's actually how receptor proteins work. A hormone will attach to the receptor and it won't actually enter the cell. The receptor will send a signal inside to then cause a particular reaction to happen. The fifth thing, the last one, is glycoproteins. So glyco has to do with sugars. We're going to learn a new type of sugar now. We've learned, remember, polysaccharide, monosaccharide, disaccharide. In this case, these are called oligosaccharides. Oligo means a few, because when we've talked about polysaccharides, we're usually talking about like starch and glycogen, and these are like 100 sugars long, really, really big, bulky things. Oligosaccharides are just like a little short chain, but these are really important because these are ID markers. And that kind of a protein, for example, determines your blood type. If you have a marker that's called A, your blood type is A. You have this actual marker on your red blood cells. If you receive the wrong blood type, that marker will attach to basically the incorrect blood that's given to you. You make antibodies. It causes this whole clumping reaction. It's also the reason why you couldn't just donate a kidney to anybody, because these surface markers identify your tissues as belonging to you. They're going to be more similar in family members, but at the same time, that doesn't necessarily guarantee you could donate a kidney to your sister or anything like that. Another thing that'll happen with these glycoproteins, they can be attached to things like in your immune response. You have certain white blood cells that will actually tag bacteria with glycoproteins that basically mark them, like put little bullseyes on them, sort of, so that the other types of white blood cells will then turn around and attack them. So ID markers can identify something as belonging, can also identify something as not belonging inside of the cell. Okay, and this is just a diagram. We went through all of these: transporting stuff, enzymes, attachment, glycoproteins, joining cells, and then receptors. Finally, to finish this up, what can and can't pass through the membrane. So I mentioned this before, lipid-soluble things can go right through. Also small things like carbon dioxide, oxygen, and water. Now water's polar, so you would say, well, I don't understand why water can pass, because you said earlier that polar things shouldn't go through. Lipids, water, they would reject each other. But water, there's actually little channel proteins called aquaporins, and water flows freely through those. So carbon dioxide, oxygen, water, and then lipid-soluble molecules are easily passed through the membrane. That's why toxins that are lipid-soluble tend to get stored in cell membranes, and you have a harder time getting rid of them. Things that can't pass would include large polar molecules, like sugar, for example, And then ions, hydrogen ions, sodium ions, chloride, magnesium, calcium, and these are all ions that cells need for certain jobs. So this would be really important, that you would need some kind of proteins to carry these things across if the cell was going to need them. Alright, this is part 2 from our membranes chapter, and in this section we're going to talk a little bit about transport across cell membranes. So we're going to start with the simplest, and then we're going to work our way towards some of the more difficult concepts. So the first one we're going to talk about is diffusion. So diffusion is really simple. It is just the movement of molecules from a higher concentration to a lower concentration. And it's really important, really important, that you understand that this does not require energy because molecules are always moving. So as molecules bounce around and they hit each other, the collisions are going to cause them to go off in random directions, and what's going to end up happening is if there's more molecules in one area, they're going to be more likely to hit each other and get bounced around, and eventually what's going to happen is the concentration of molecules is going to diffuse. You're going to end up with similar concentrations all over the place on both sides of the membrane, not because they're going in one direction on purpose, but just because of random motion. And osmosis is just a special name for the diffusion of water, because water is so important. When we're talking about diffusion, we're usually talking about solutes, ions, covalent molecules, etc. When we talk about osmosis, now we're talking about a solvent, because a lot of the time the solutes are stuck where they are. But the solvent, water, can freely flow across the membrane, because if you recall, we have those aquaporins that allow water to freely flow through the membrane. Okay, so this is sort of showing a diagram. At the top we have molecules of dye, and what they're showing is that the dye is bouncing around randomly, but every time it hits a space, a pore in the membrane, it passes through. It's not that the dye can't go the other way. If we look at the second slide here, you see that some of the dye molecules are going the other way, but the chances of one hitting from the left are much higher than the chances of one hitting from the right. Eventually, once the dye molecules have spread out, the chances of one hitting from one side are equal to the chances of hitting from the other side. And so what we have is what you really should call dynamic equilibrium. It means that the concentration is the same on both sides. It's not the exact same molecules on both sides, because I gave the example in the classroom that we have two doors. If, let's just say, everybody was sort of bouncing around inside the room and there was also people outside the room, and every time somebody from outside got knocked in a door, somebody that was inside the room got knocked out of a door, we would maintain the same number of people in a room, because every time somebody went in, somebody else would go out. However, it wouldn't be the same exact people. So it would be called dynamic equilibrium, the fact that the molecules are still moving, but the actual concentrations aren't changing. So that's diffusion. Now, tonicity is a really important concept that we're going to talk about, and I mentioned this a moment ago, the fact that a lot of times the solute molecules cannot pass through the membrane. It's the water that can pass. And so this plays a huge role in equilibrium in a cell. So here's sort of your keywords. Hyper means more, hypo means less, iso means equal, and tonic is going to refer to the concentration of solutes. So the more concentrated one side is to another, that's going to be the hypertonic side. The side with a lower concentration of solutes is going to be the hypotonic side. And you can't have one without the other. I couldn't just give you a picture and say, hey, is this a hypertonic solution or a hypotonic solution? That would be like saying, is she smarter? You have to have a comparison. Is she smarter than who? And so the same thing here. You'd have to have two sides to compare to one another before you could say that one side was hyper or hypo. Now the rule, and you can just memorize this, is that water's always going to go towards the hypertonic side. So if you can figure out which side is hypertonic, that's going to be the way the water's going to go. And that's because if there's more solute, then proportionally there has to be less water. I'll show this to you in a diagram in a second. So let's start with the simplest one, isotonic. Isotonic is a situation where the concentration of solute on both sides is equal. It doesn't have to be the same solutes. If the solute is unable to pass across the membrane, then 20% of one kind of solute on one side is going to be the same as 20% of some other kind of solute on the other side. Because if 20% is solute, the other 80% is water. So that means both sides have an equal concentration of solute in comparison to water. Okay, so here's an example of what I'm referring to. Let's just pretend like right here is some kind of a semi-permeable membrane separating two sides. So on the left side of this semi-permeable membrane, I have a 20% sugar solution, and on the right side, I have a 20% salt solution. So that means that there's going to be these little sugar molecules bouncing around, and chances are that if one of these little sugar molecules hits this membrane, if the membrane was permeable to sugar, then the sugar would go through. And same idea that if on the right side of this membrane we have 20% salt, so here's our little salt molecules, and if a salt molecule happened to hit this membrane and the membrane was permeable to salt, then the salt would go through. But let's pretend like the salt and the sugar, that the membrane's impermeable to both of these. So anytime salt or sugar hit this semi-permeable membrane, they can't cross. But what we do know is, let's just say that this membrane is freely permeable to water. The reason I say this is an isotonic solution is because if water can pass, a solution's made of two things, solute and solvent. So if this left side is 20% sugar, the other 80% is water. And the same is true over here. If this side is 20% salt, the other 80% is water. Therefore, if the solutes are stuck where they are, the amount of water, the concentration of water on each side of this membrane is the same. And it doesn't mean everything stops, it just means that the speed at which water is going in one direction is going to be equal to the speed at which water is going in the other direction, and so we have what's called dynamic equilibrium. And that would be the situation in an isotonic solution. Okay, so if the solutions are not isotonic to each other, then that means that one side is going to be hypotonic, meaning it's going to be the side that has less solute, and the other side is going to be hypertonic, which is going to be the side that has more solute. Remember that if there's more solute, I just showed you this, that means there's less water. And therefore, water's always going to go from higher concentration to lower concentration, so you can kind of just memorize that water's always going to go to the hypertonic side. So let me show you an example of this. Okay, so at the top of this I've got 3 beakers, and each beaker has we're going to pretend like this is a cell. And the cells, each of the cells has 10% salt solution. And then outside the cells we have different concentrations of salt in the solution. So in our first solution we can see that it's 10% salt inside and 10% salt outside. And assuming salt can't pass, we have 90% water on each side, so this would be isotonic. In our second situation, we see that we have 10% salt inside, but we only have 2% salt outside. So that basically means that outside we have 98% water, and inside of our cell we have 90% water. Now, remember that water is going to go from where there's more water to where there's less water. And so you can actually look at this and say, okay, that means water is going to go into the cell because there's more water outside than in. However, as I mentioned to you before, if you just look at the solutes, determine which side's hypertonic. In this case, inside has more solute, so the inside is the hypertonic side, and the outside is the hypotonic side. Water's always going to go towards the hypertonic side, because by definition that means it has more solute and therefore has less water. If you look at our third problem, we have the opposite situation. Inside the cell 10%, that means that it's 90% water. Outside the cell 30%, that means it's 70% water. Again, if we just look at tonicity, the outside is hypertonic. The inside, this time, is hypotonic. And so water, in this case, the net movement would be out of the cell. The second problem here is what you see a lot of times. This is called a U-tube problem. So what they do is they have this U-tube with a semi-permeable membrane. They always have to tell you what can pass and what can't pass, and then they're going to ask you questions about this. So the first question here is to describe A versus B at the beginning. To do that, all you've got to do is just look at A and B and total up the solutes on each side. So if we add up the solutes on the left and we add up the solutes on the right, we're going to see that each side has a solute concentration of 3 molar. And so therefore, the answer to this first question is that at the beginning of the experiment, it's isotonic. Now here's a second kind of question they can ask you. Describe side A to side B with respect to glucose. So this time they specifically want you to just look at the glucose and ignore everything else. So if we do that, you're going to see that side B has more glucose than side A. Therefore, side B is hypertonic with respect to glucose. The second thing says, or the third one, if sucrose can't pass but glucose can, which way will it go? Okay, that's our standard definition of diffusion. Anything that can pass is going to go from where there's more of it to where there's less of it. So glucose is going to go towards side A because that's where there's less glucose. And now the hardest question, which way will water go? The way to answer this question is look at the solutes first. They're going to give you something that's pretty easy to figure out. It's not going to be something where you have to use math in order to figure this out. At the beginning of our experiment, it was isotonic, so that would mean that water would be going in both directions at the same rate. However, if we know that the glucose is going to go to the left, then that means that what's going to happen is that our left side, side A, is actually going to become hypertonic to side B. So if glucose goes towards this side, that's going to mean that the concentration is going to go up on this side. The concentration of solutes is going to go down on this side. And that basically means that water is going to go towards the hypertonic side, which is going to be side A. And what we would see at the end of this experiment would probably be that the water level would be higher on side A and lower on side B. So that's how you solve one of these YouTube problems. Alright, so now that we've discussed the fact that water can freely flow into and out of cells, because remember they've got these aquaporins that let water through, we know that what's going to happen is if it's not an isotonic situation on both sides, water is going to either be entering a cell or leaving a cell, and that obviously is going to cause a problem because cells need homeostasis. So osmoregulation is basically the term for maintaining water balance or controlling the water balance in an organism. So let's start with freshwater. In freshwater, you have organisms that are going to be hypertonic to their environment. In other words, they're going to have more solute in their cells than freshwater is going to have on the outside. So the paramecium is an example we talked about in the last chapter. Paramecium has a special vacuole called the contractile vacuole, and it basically just squirts out excess water. Freshwater fish make lots of urine. That's their way of getting rid of it. It actually works to their advantage because one of the waste products they make is ammonia, and ammonia is highly toxic. And when we make ammonia, we have to expend energy to turn it into urea, which we then excrete in urine. Fish don't have to use energy to do that because since they have a constant water supply, they can just constantly flush the ammonia out of their bodies. So by making lots of urine and by they can basically survive and use it to their advantage. Saltwater fish have the opposite problem. They're basically living in an area where the area would be hypertonic to them. The way that they solve this problem is, number one, they maintain high solute concentrations within their body, because remember, the solutes don't have to be the same, just the water concentration. So they typically maintain high urea concentrations to match the high salt concentrations on the outside. Another thing that a lot of them do is they actually expend energy excreting salt. And we see this even in, like for example, saltwater crocodiles who have to drink salt water. They have salt glands to get rid of excess salt. Okay, in plant cells it's a little different. Plant cells have cell walls and central vacuoles. So what they're going to do is they're going to basically absorb as much water as they possibly can. It's going to create the pressure that they need on their cell wall that actually maintains turgor pressure, meaning it's what makes the plant leaves and stem and all of that stiff. unlike animal cells that are normally in isotonic solutions, plants are typically, their cells are typically hypertonic to the external solution because fresh water is constantly coming in at the roots. And in fact, if a plant cell is put in an isotonic environment, it will probably get somewhat wilted, which the word for that is flaccid. Extreme condition would be if you were to put a plant in salt water or under severe dehydration. If enough water is lost, what will actually happen is the cell membrane will pull away from the cell wall, and that's called plasmolysis. And we saw that in the lab with the Elodia. It will kill the cell. Okay, so thus far everything that we've talked about has occurred without energy, just due to the random motion of molecules. The rest of this lecture, for the most part, is about the type of transport that requires energy, and in general, that's called active transport. Anytime movement is occurring from low concentrations to high concentrations, in other words, upstream against the gradient, the only way that's going to happen is if the cell expends energy to cause it to happen. Sort of like if you have a leak in a boat, the water's going to come in without any energy. That's with the gradient. But on the flip side, if you don't want to sink, you're going to have to expend energy to pump water back out so that you stay afloat. So when we see active transport, we're going to see that ATP is usually involved, and that means you're going to see mitochondria in areas where active transport is occurring. So in this little animation, it's just showing how the phosphate actually breaks off of the ATP, and you see that it now becomes ADP, which is adenosine diphosphate, and the phosphate itself is what causes the conformational change in the protein. In this animation, one thing I don't like is that it implies that, in this case, it looks like it's going from higher to lower. And generally, we're talking about things going in the opposite direction, lower to higher, when we're talking about active transport. So the textbook example of active transport is a protein known as the sodium-potassium pump. And I have it diagrammed here. It's from your book. And it basically shows that what happens is sodium attaches to the protein. It actually 3 sodium ions attach to this membrane protein, a carrier protein, from the inside of the cell. And when 3 of them, they're attracted to it, when they attach to it, the next thing that happens is ATP comes in and drops off its phosphate. When the phosphate binds, that is what actually stimulates the protein to change its shape. And when it does that, you see that the sodiums are no longer attracted to it, so they're released. Potassium now comes in, and the potassiums themselves actually cause the phosphate to break off. And when the phosphate breaks off, the protein undergoes a conformational change back to its original shape, and the potassium is now released because it's not attracted to the protein anymore. One thing I don't like about this little animation is that it gives the appearance that the sodiums and the phosphates are there lined up like it's a carnival ride and they're waiting to get on, and that's not what's happening. We still have random motion of molecules, but because sodiums are attracted to it, when a sodium bumps into it, it sticks. And so what's happening is a cascade of events, sort of like a domino effect, but it's not that the sodium and potassium are lining up waiting to go through. Okay, and this next slide is sort of a summary of what we've talked about. You see on the left all of our passive transport processes. We have diffusion, simple diffusion through the membrane. Then we have facilitated diffusion, which is where things go through the membrane, but with the aid or facilitation of a protein. It could either involve a protein that almost forms like a channel, or it could be a protein that when the molecule binds to it, it sort of causes the protein's tertiary structure to change, so the molecule is sort of carried through. But none of these require energy. Over here, active transport. When you see active transport, usually you're going to see a diagram showing molecules going from where there's more to where there's, where there's less to where there's more. Like here, the little, this is supposed to be the little potassiums, and you notice there's less here, and they're going out in this direction. The sodium's also going from where there's less to where there's more, and you're always going to see something showing energy, ATP, something that's basically facilitating the motion. As concentration differences. So we have the chemical difference, the concentration that's different, but in addition to the chemical difference, we also have the electrical force, the voltage difference. So opposites attract. We have positive charge being attracted towards the inside of the cell. We also have sodiums being attracted towards the inside because there's more outside than inside. So what happens in the case of ion pumps is that they're setting up this, I guess, electron potential. Any kind of protein that generates voltage is called an electrogenic pump. So the sodium-potassium pump would be an example of this. And another good example of this would be proton pumps. The mitochondria inside our cells actually contain a proton pump where they pump hydrogen across a membrane. Hydrogen ions are protons. Hydrogen is number 1 on the periodic table. It has 1 proton and 1 electron. When hydrogen forms an ion, it's H+. That means all it is a proton. So when they say proton pump, what they're really talking about is hydrogen ions. And it kind of makes sense that mitochondria have a proton pump because mitochondria, remember our whole endosymbiotic theory, that they were bacteria. So this would be yet another piece of evidence that would support the endosymbiotic theory, the fact that mitochondria use a proton pump, which is something that bacteria use. All right, and this little diagram is actually showing what's called an action potential. You're not going to be tested on this right now, but I just wanted you to see that what you have in your nervous system, and this is sort of the whole idea of how your nervous system works, is that you have these gated channels with sodium, more sodium outside than inside, and this is sort of set up by the sodium-potassium pump. And what happens is when a nerve gets stimulated, actually it starts down here, when a nerve gets stimulated, the sodium gates open, and sodium starts flooding in. Why? Because number 1, the positive charge is attracted to the negative side, and number 2, you have a higher concentration of sodium on the outside than the inside. And when that happens, it basically causes a domino effect, and this is basically your nerve impulse that's going to travel down the neuron on its way to stimulate the spinal cord, go to the brain, stimulate a muscle, wherever that particular nerve impulse is headed. So, the next thing here is co-transport. Now, co-transport is where you have a molecule, and for example, in this diagram, you have sucrose. Now, sucrose itself does not require energy to get into the cell. But notice that what's happening is this particular protein will only carry sucrose across if, in addition to sucrose, hydrogen binds. And because it takes energy, a proton pump, to pump the hydrogen out here, then indirectly, sucrose does require energy in order to get into the cell, because the hydrogens wouldn't be out here without energy going in to pump the hydrogens out there. So this is co-transport. This is where a particular molecule that a cell needs can only be transported in when there's a second molecule attached to it. And in this case, it's hydrogen, but the idea is in co-transport is that it's an indirect type of active transport because the molecule itself doesn't need ATP to be transported, but it needs something that ATP was required to get to its location, if that makes sense. Okay, and another type of active transport is called bulk transport, and this is where something bulky, something really large, is taken in or out of the cell. So this is something that's too big to actually just go through the cell membrane or to go through even a protein channel or a protein pump. It actually requires the cell to wrap around it in order for it to get where it's going. And so this also is going to require energy. So endocytosis is the general word for a cell taking something in by the cell itself, actually the membrane wrapping around it, surrounding it, and then bringing it into the cell. Phagocytosis is specifically cell eating. This would be, if you were going to see this in a diagram, the little vesicle that forms around it would be around one giant bulky thing, like a bacteria, like an endosymbiotic theory. We were looking at phagocytosis there. The second kind is called penocytosis, which is cell drinking, and what you'd see in that case is the vesicle forms around a bunch of little things. And the last type is called receptor-mediated endocytosis. And receptor-mediated endocytosis is where the only way the thing can get in is that it has to bind to a receptor, and then the receptor stimulates the cell to then take in the particle. So cholesterol is a really good example of something that is taken in by receptor-mediated endocytosis. And exocytosis is basically just the opposite, the cell getting rid of a particle or something like that. Here's phagocytosis, very clear here, big thing. Here, penocytosis, a bunch of little things. And this is receptor-mediated endocytosis. This could be cholesterol, for example. The cholesterol has to bind to receptors, and that stimulates the cell then to take it in. And so you could have, for example, eat the same diet as somebody else, but maybe your cholesterol runs high, and it's because these LDL receptors that pick up particularly low-density cholesterols, which are the bad ones, maybe you have less receptors genetically. Now, you can't pick up cholesterol and remove it from your blood and process it like somebody else that eats the same diet as you. The last section of these notes is about cell